Admit that every time I see John's presentation, I don't know whether to be giddy with excitement at the opportunities or run screaming from the room, but we can explore that in the question and answer period. The next presentation is Dr. Lansing Taylor from the University of Pittsburgh. Lans is also an active member, participant, contributor to the NCAS Tissue Chip Project, and that's where we've had the good fortune to work together. And he's going to explore quantitative systems pharmacology and the bridge from in vitro to in vivo toxicology and disease modeling.
Thank you, and I'd also like to thank the committee for inviting me to talk about QSP and the bridge from in vitro to in vivo tox and disease modeling. My agenda, at least my goals going last in the series of speakers was to see if I could bring a few things together. So I'm going to talk about some of the key challenges in developing precision therapeutics, application of QSP to advance the development of precision therapeutics, developing and implementing a human liver microphysiology platform to investigate both ADMET and disease modeling, the critical role of human iPSC-derived cells for drug discovery and development, and liver diseases investigated in the liver MPS. So animal models obviously continue to be valuable, yet they do not always exhibit concordance with human mechanisms of disease and toxicology. So therefore, there needs to be a complementary approach. In addition, the variability in the genetic makeup of patients demands a human precision medicine approach to developing and testing new drug candidates, and this is where the iPSC technology comes in, with the goal, as we've seen in a couple of talks this morning, of getting individuals on a chip for drug testing and for exploring mechanisms of disease progression. So we know humans are complex, heterogeneous systems, and this is obviously a challenge in drug discovery and precision medicine. We're well aware of the heterogeneity within a population. However, if we think about an individual patient, there's heterogeneity in the system going from organs to tissues to cells, the major components that make up the cells, where the heterogeneity is really driven primarily by mutations. And this is a challenge, of course, in some of these molecules are targets for drug discovery. We also know that these and other components interact in time and space to bring about normal functions, or in disease, abnormal functions through pathways. However, this is a qualitative characterization of the interactions, but it's possible to use network theory, a branch of mathematics, to build quantitative, computational models of these interactions. The value of building these computational models is that you can make predictions and then go to experimental systems to test the predictions and then update the models. Clearly, we're interested in adding to the experimental modeling component with human experimental models using especially microphysiology systems. And again, taking advantage of patient cohort iPSCs, and this data can be compared to animal models of the disease. So we believe we can advance precision drug discovery through the application of quantitative systems pharmacology, or QSP. We start with a focus on the patients, and then we can take advantage of the data from electronic health records, patient clinical samples, the ability to perform a variety of omics on those samples, and then warehouse that data for further analysis computationally. But for precision medicine as practiced today, in getting the right drug to the right patient at the right time at the right dose, we can use the predictive capability applied to this data to make those decisions. However, that's based on existing drugs today. We think we can continue this circle by applying therapeutic area basic science, adding more sophisticated experimental models of disease coupled to the computational models of disease in order to identify novel therapeutics. And this is what we would call QSP-driven drug discovery. So a functional definition of QSP, and there's been lots of definitions over the last 6 or 7 years of the evolution of this field, Our functional definition is determining the mechanisms of disease progression and mechanisms of action of drugs on multi-scale systems through iterative and integrated computational and experimental methods to optimize the development of therapeutic strategies. So we've implemented at the University of Pittsburgh a whole platform. So all the disease we're studying in the Drug Discovery Institute, we go through this process. Again, starting with patients and patient samples. We can also take advantage of validated target knowledge. For example, in metastatic breast cancer, we know that mutations in the ligand binding domain of the estrogen receptor does play one of the roles in metastasis. But we can also take an unbiased approach, for example, doing RNA-seq on primary tumors in the patient, as well as metastatic tumors in a breast cancer patient, and then infer the pathways of disease progression using the computational methods that are available. This then gives us a list of potential molecular targets, those components that are in those inferred pathways. We can then take those list of targets and go to databases like DrugBank and Stitch and use machine learning tools like latent factor models and predict existing drugs that might interact with that list of targets. So now we have the potential to start a pathway to repurposing drugs and also the starting point for probes to develop new drugs. A key step here is building, in the way we implement this platform, phenotypic models of disease and safety. And the goal here is to create disease models that are sophisticated with the goal of looking for molecules that will reverse the disease phenotype. We can also use tools to manipulate the models, RNAis, CRISPR, mutant cDNA libraries, as well as some focused compound libraries. We use high-content screening in these models, particularly the MPS, in order to analyze the various phenotypic functional changes. Because it's an image-based system, we can actually quantify the heterogeneity that is always present in biological systems. We can then identify the drug hits from the profiling, do medicinal chemistry just based on the phenotypic results. But we also want to know what the molecular targets are, so we identify those highest probability targets using chemical proteomics and RNAi technologies. If we have a small enough list of probable molecular targets, we can revert to a target-centric drug discovery process. We also build mammalian models of disease and safety. And with all the data that's in the literature and from the experiments from the animals and the in vitro studies, we can build the initial computational model. This is iterative, so we have to go back around the scheme because we're going to make predictions from the initial computational model. We can go back to the experimental model and knock things down, change things, and generate new data. This is a systems biology approach, so we expect some emergent properties, like optimal therapeutic strategy, PD biomarkers of drugs, and prognostic and predictive biomarkers. We also believe we can be involved in clinical trial design and simulation, and as was already brought up, the potential to use the microphysiology systems in essentially a preclinical trial perspective. So I'll give my view of the status of the development of human MPS. Over the last 5 years, there's been tremendous progress. On the other hand, no MPS yet has recapitulated all the cell types in any organ, but significant functions have been demonstrated and are already useful as experimental tools to understand disease progression and to predict drug pharmacology, metabolism, disposition, and toxicity, where we're able to have full experimental flexibility in the system. Many organ MPSs are in development, but more need to be initiated and compared to animal model data. The measurements made in MPS are often different than in animal GLP tox studies, but important functional and structural measurements can address specific questions. Physical coupling, as you heard from John, of organ MPS is a major physiological challenge but is in progress, and there's been some initial success. The critical development of iPSC-derived cells to construct MPS has progressed dramatically, yet requires more development for full maturation and validation. And the validation of the MPS models themselves needs to be demonstrated, especially the reproducibility, and that's now underway at tissue chip testing centers using some of the computational tools that we've developed. So in entering the use of MPS, you have to make some decisions on what are the most important things to do. Our list includes testing human ADME tox and disease models, including a high throughput version, which I'm not going to talk about today, and a lower throughput mechanistic platform, the MPS. Testing the role of liver zonation in toxicology, disease initiation and progression, creating a human liver metastatic site to investigate cancer cell extravasation, cancer growth, immune infiltration, and drug responses, and creating a human liver disease model for the NAFLD spectrum of diseases. This shows the platform that we've developed for actually interrogating the MPSs. In the upper left, we see the newest generation liver MPS, the so-called vascularized liver acinus MPS. It consists of all primary human cells obtained from the same patient, hepatocytes, endothelial cells, stellates, and Kupfers. We also are making iPSCs, and we replace one of the primary cells when we've made significant progress. We also have sentinel cells. This is usually 10% up to 20% of the cells where we incorporate a fluorescence-based biosensor, for example, for ROS production or apoptosis. The system is under continuous flow, so we can monitor the efflux media, looking at secreted molecules, measure oxygen pH, and we can measure metabolic readouts, for example, of drugs and drug metabolites using mass spec. On the B, on the bottom line, we use high-content imaging to make the optical readouts. All of this data is collected into the microphysiology systems database, designed to acquire, manage, analyze, and ultimately model the data from the MPS, including the critical metadata, so that reproducibility can be defined based on how things have been done. It's not working. So we've defined the building of MPS systems as an evolutionary process. Our first generation system was the sequel cell, or the sequentially layered self-assembly liver model. Used primary human hepatocytes and then cell lines, human cell lines for the other cells, where we demonstrated the functionality of those cell lines. 3D structure came from sequentially layering the cells to recapitulate the liver acinus. The first generation had only zone 3 oxygen. We had a few biosensors and a simple secretome. The next generation system, the liver acinus MPS, we increased the number of primary human cells, added to the three-dimensional structure by adding a space of DC between the endothelial cells and the hepatocytes. And on purpose, we created a zone 1 and a zone 3 model so we could explore zonation effects. We added to the biosensors, added to the secretome. The last generation, the system we're working on now, is designed for all human iPS cell systems. Today, we've started as a validation, as a reference point, all primary human cells. Three-dimensional structure comes from actually recapitulating the liver sinusoid. We have a vascular chamber separating from a hepatic chamber through a PET filter with 3-micron pores, and we have separate control of flow in the vascular chamber versus the hepatic chamber. We create a continuous zonation. We're now gene editing iPSs, and we're increasing the secretome that we're measuring. So the holy grail is to create a full biomimetic of the human liver acinus, and this shows the classical diagram where we have on the left blood flow coming into the liver sinusoid. It's at high oxygen tension, which is zone 1. Primarily, hepatocytes consume the oxygen as it flows through the sinusoid. So before it enters the central vein, it creates a zone 3 within the liver acinus. And so therefore we have kind of a continuous change in the microenvironment of the liver. Below shows some of the characteristic measurements made primarily in animals that show a bias to activity in zone 1 versus zone 3. So the goal was to recapitulate that. And so with the LAMPS model, we engineered 2 different zones. Zone 1, which we engineered to be at high oxygen tension, and then zone 3 at a lower oxygen tension. We computationally modeled that, knowing the diffusion of oxygen in the materials we had, the oxygen in the medium, the consumption rate of oxygen of the cells. And in D, the gold points show the computationally modeled oxygen tension within the zone 1, and the blue dots are zone 3, and the red diamonds are actual physical measurements of oxygen tension using a ratiometric method we developed using a oxygen-sensitive fluorescent probe. We then wanted to define the biology from a zone-specific state, and zone 1 is in red, and zone 3 in blue, and in the left-hand column you can see what was expected to be greater in zone 1 than in zone 3. That's albumin, urea secretion, oxidative phosphorylation, and glucose concentration. In the right-hand column is what was expected to be greater in zone 3 than in 1, and that included alpha-1 antitrypsin secretion, CYP2E1 expression, steatosis, and acetaminophen toxicity. So we also have additional measures where we demonstrated CYP's functions and phase 2 conjugation in the models. Positive control drugs induced expected hepatotoxicity. We were able to induce methotrexate-induced fibrosis and immune-mediated hepatotoxicity. So now we have an experimental MPS liver model to explore the role of zonation in disease as well as in ADMET. So this just shows some of the data we've generated. We also have a collaboration with EPA. It's a collaboration also with John Wixwoe at Vanderbilt. This just shows some of the drugs that we've tested. On the right is the key, and I'll just zero in on methotrexate, which we were able to demonstrate fibrosis as a key. And this just shows some of the data at endpoint measurement after 21-day exposure to methotrexate at 30 nanomolar. On the left in A is a negative controls, and the stellate cells we engineered to express a green fluorescent protein. And when we labeled for an antibody to alpha SMA, there was very little labeling, very little within the hepatocytes where you can see the red, but none within the green stellate cells, or they'd appear yellow. In contrast, in B, in the presence of 30 nanomolar methotrexates, the stellate cells that are green now appear yellow because of the fluorescence from the SMA antibody. We can also see that there is increased fluorescence of SMA in hepatocytes, and this has been debated in the literature for a long time. We see it in our in vitro models. We did a separate experiment where we labeled after 30 nanomolar methotrexate for 21 days for collagen, and again we see the green stellate cells now have expanded, and they also have collagen 1A2 in and around the stellate cells. So we've, the last generation system is the vascularized liver acinus, and this allows cancer cell extravasation, immune cell infiltration, and delivery of factors from other organs. It's primarily made of glass and plastic, minimal polymer, and this is crucial because although PDMS is a great material for engineering devices, they're sponges for hydrophobic molecules like drugs. This shows a diagram in A where we kind of have the separate vascular chamber and the hepatic chamber. In B, that just shows the device. C, the continuous zonation in the device. And D and E shows functionality within the device. This happens to be one we've started on the Naffeldy model. So in E, we can see early simple steatosis. The system is scalable. We can clamp large numbers of these devices in a single device. We can separately perfuse them, and we can fill incubators with those. So we're now running a couple of dozen of these in parallel. I mentioned the iPSCs, and since that has been discussed already, I won't go through it, except to say that we're now engineering those. And for the Naffeldy project, we did a proof of principle. We know that SIRT1 is involved in steatosis, and so we have a control where we have a doxycycline-controlled knockdown of genes. In the absence of DOX, we get normal hepatocyte structure. When we knock down SIRT1, we get macrosteatosis. We've also engineered a rat as a bioreactor, so we can now mature the hepatocytes to essentially 90% adult function. In terms of creating a model of non-alcoholic fatty liver disease, using the conditional control of knocking down specific genes, using SIRT1, we can also add products from the intestine and fat, as well as circulating immune cells through the vascular channel of the device. And again, given the fact that I'm now over, correct? I'm blinking red.
Yes, you are.
So I'm just going to skip to the end and say one of the things we're doing in metastatic breast cancer is building a model that we hope will be a complement to the PDX mouse models. So the first set of experiments was to take MCF7 cells, this is a cell line, human cell line that's ER positive, and we produced mutations in the ligand binding domain, making 2 mutants. And one of the things we did was to compare the growth characteristics in 2D culture, in a liver cell culture, as well as in the microphysiology system. We engineered the cells to express a fluorescent protein so that we could monitor growth and the response to drugs. The bottom line here is in 2D culture, one of the mutants, the D538G, actually has a growth advantage. However, when we move in the center to co-culture with the liver cells, there's a switching. The second mutant becomes dominant in terms of growth, and we move to the MCF7 in the LAMPS model. That switch remains, but the overall growth rates of all the cells are depressed. I might add that we're in the middle of a bunch of experiments now looking at extravasation of cancer cells through the vascular system. We're also doing drug testing because we don't have to worry about the PDMS. So in closing, we think the future of QSP and MPSs are strong. We think it's important to implement the full QSP platform for liver diseases, starting with the patient samples and data, develop multi-scale experimental and computational models of liver diseases for drug discovery, along with PK/PD for drug development, and the key role that human iPSCs will play. Final slide just is acknowledgement of the large number of people that have helped our work. Thank you.